Hello, everyone. In today's demo session, I will explain to you how to perform the data analysis for a qPCR experiment. So to do this, I have provided the following Excel file, where in the first column, you can see different sample numbers. So the samples are numbered from 1 until 30. And these samples were derived from Arabidopsis thaliana plants exposed to different cadmium concentrations. And you can see in the second column here, titled exposure, which cadmium concentration the plants were derived from. So as you can see, there are 6 so-called biological replicates per experimental condition. So we have 6 control samples, 6 samples derived from plants exposed to a concentration of 1 micromolar of cadmium, 6 samples derived from plants exposed to 2.5 micromolars of cadmium, 6 samples from plants exposed to 5 micromolars of cadmium, and finally also 6 samples derived from plants exposed to 7.5 micromolars of cadmium. And then in these samples, I measured the expression of different genes. The first one is SMR4. This is a gene that encodes a protein which is involved in cell cycle regulation. And in this case, this was my gene of interest. Of course, normally you will probably measure more than just one gene of interest, but for the sake of simplicity, for this example, I will perform the calculations for just one gene of interest, but the main mechanism is exactly the same for every gene of interest that you want to analyze. And then here in columns D until M, you can find the CQ values that I obtained in the same set of cDNA samples for 10 different reference genes. So all these genes listed here, these are the reference genes that I want to validate in my experiment. And this is immediately the first step of the data analysis, which is that we will first determine which of these 10 reference genes are most suitable to normalize the expression of the gene of interest, which in this case is SMR4. So what I will do is I will create a new tab in my Excel file, and I will name it reference genes. And here I will already copy the first 2 columns with the sample numbers and the corresponding cadmium concentrations. I will zoom in a little bit. And then next, I will also paste the CQ values from the different reference genes. So don't copy the CQ values for SMR4, only copy the values for the 10 reference genes. So I will copy these here. And for the sake of clarity, I will put a column on top, which I name, or a row on top, which I name CQ, so that you know that these values are the obtained CQ values. Now, what we will do in a first step of selecting the appropriate reference genes is that for each of these columns, we will determine the lowest CQ value. So we do this for each reference gene individually. So I will place here in bold minimum CQ, so the sample with the lowest CQ value. And as you will hopefully recall from the lecture on qPCR principle, the sample with the lowest CQ value for a certain gene is a sample which has the highest expression of this gene, because the CQ value and the expression level are reversely proportional to each other. So we will now determine the minimum CQ value for each reference gene. And we simply do this in Excel by using the formula for the minimum, which is the equal sign followed by min. Then we open the brackets, select the entire column of CQ values, and then close the brackets, and then press enter. And here you will now find that the lowest CQ value for this reference gene is 23.18, more or less. And when I then have a look, it seems that this one corresponds to sample 23. So for this reference gene here, the sample with the lowest CQ value and hence the highest expression of the gene is sample 21. Now we need to do the same calculation for all the other reference genes as well. Of course, you can always fill in the formula again, but what you can also do in Excel is just select this value, click the bottom right corner of this cell, and then drag it to the right. And then Excel will perform the same calculation for each of the columns. So we now know the lowest CQ value for each of these reference genes. Now the next thing that we need to do is determine the so-called delta CQ. And the delta CQ is the difference between each of the individual CQ values and the lowest CQ value. So that's what we will do next. I will just copy this here and I will name this delta CQ. Insert a delta sign. Voila. So what I will do for the first reference gene is I will fill in the formula equal sign this value minus the minimum CQ value for this reference gene. And then we can see that the difference in CQ value is approximately 1.19. Now we need to do the same thing for each of the samples in this column. However, if I now drag this formula down, the problem is that it will indeed in this cell calculate the difference between this cell here, so the CQ value of sample 2 for the specific reference gene. However, it will then no longer subtract this minimum CQ value from it, but it will also move a cell down. And this is of course not what we want, but to avoid this in Excel, we can make use of dollar signs. A dollar sign in front of either the row or the column means that you fix that row or column. If you add a dollar sign both in front of the row and the column, you fix the entire cell because you will fix row and column. In this case, we need to fix the row. So we want in our formula that it not shifts down from row 34 to 35 and so on, but it will always use the value in row 34. However, the column can shift because then we can drag the formula to the right to do the same calculations for the other reference genes. So this means we need to put a dollar sign in the formula, but we only need to put it in front of the row. The row is number 34, so we put a dollar sign here. So the formula becomes cell D3 minus D dollar sign 34. Now I can drag this formula down. And you will see that the calculation was done correctly. And you can verify this by the fact that for one of the samples, you should obtain value 0. As you remember, the sample with the lowest CQ value was sample 21. So of course, when you subtract that value from itself, you will obtain 0. Now we will do the same thing for the other reference genes, and we can simply do this by dragging the formula to the right. So I'll do it like this. So now we calculated the delta CQ, which is for each sample, the difference in CQ value between that sample and the lowest CQ value of all the samples for that same reference gene. What we will then determine next is the so-called relative quantities. So we abbreviate this as RQ, and we will again do this for each sample, for each reference gene. And these relative quantities are calculated using the so-called 2 to the power of minus delta CQ method. The delta CQ is what we just calculated. So this actually means that we calculate the difference in expression between each sample and the sample with the highest expression. So these are the values that we obtained here. Now we will calculate the so-called RQ values. So as I said, the formula we will use is equal sign 2. Then we take the power of the negative value of this delta CQ. So 2 to the power minus delta CQ. And then we press enter. And then we can again drag this formula down. And we can also drag the formula to the right. And you can again verify whether the calculation was done correctly, because now in each of the columns you should be able to find one value which is equal to 1. And this is the sample with the highest expression, because of course if you calculate 2 to the power of 0, you will obtain 1. And basically what these data tell us is, for example, for the first reference gene, so AT4G26410, we can see that number 1 is found for sample number 21. So this is the sample with the highest expression, and now we expressed this highest expression as value 1. All the other values in this column are expressed relative to the expression in this sample, so relative to 1, which means, for example, that this sample here, which is sample 5, it has an expression of 0.5. So this means that the expression of this specific reference gene in sample 5 is half of the expression of that same reference gene in sample 21. So in sample 21, the expression of this specific gene is twice as high as in sample 5. So we now calculated these RQ values. And as you see, we tested 10 different reference genes. What we will now do is use an algorithm to determine which of these 10 reference genes, or which combination of these reference genes, is most suitable to normalize the gene expression data from the gene of interest, which is SMR4. And to do this, we will use the so-called GreyNorm algorithm. There's different algorithms that you can use, as I explained in the lecture on data normalization. Another example is GNorm or Norm Finder, but GreyNorm is the algorithm of which I explained the principle during the lecture. This algorithm was developed here at Hasselt University, so this is the one that we will use today. So what we first need to do is generate the input file for GreyNorm. And I already included a next step where I already put some of the important information that we need to include. So the first thing that you will see here are several rows, which are all preceded by a hashtag. The hashtag means that these rows give some additional information to the GreyNorm software, which it requires to perform the analysis. The first row asks you to fill in a dataset identifier. This identifier could be anything. The GreyNorm algorithm will not really use this. It's just useful for yourself if you're performing multiple GreyNorm analysis to memorize which analysis this file is linked to. So in this case, I can call it toxlearn. Then in the next row, I need to fill in the number of variables. And in this case, the number of variables in the experiment is 1, because the only variable that I included is the cadmium concentration. If in my experiment I would have also worked with different genotypes, then the number of variables would have been 2, because one variable would be the cadmium concentration, the other variable would be the genotype. Then in the next row, we need to fill in the number of reference genes that were measured. And in this case, it's 10. We always recommend measuring the expression of at least 10 reference genes, but this could, of course, be different. It could be more than 10, or in some cases, it could be also less than 10. Then the fourth row will ask you for the sample ID. And this is simply the name of the column in which you can find the sample numbers. And here I already included the first part of our data set. And here I named the column for the sample numbers also sample ID. So it's important that here and here you put exactly the same words. So you could also just name it sample or anything else, but it's just important that these two words match. Then next, we also need to list the reference genes that we measured. So these are the names of the 10 reference genes separated by commas. And also here it's important that they're written in exactly the same way as in the titles of the columns that you will include here. Because if there are small typing errors, the analysis will not work. And then the next or the last thing that the input file requires here is that you fill in which of the conditions is your control. And in this case, it's quite easy. We worked with different cadmium concentrations, but of course, we also included control samples which were not exposed to cadmium. So in this case, the cadmium concentration is 0. So here you fill in condition equals 0. Then we need to fill in the rest of this input file. So here we will put the different values that we obtained. So here for condition, we will indicate which condition each sample belongs to. So remember that we have 6 samples which were not exposed to cadmium, 6 samples that were exposed to 1 micromolar of cadmium. So I will name these 1. 6 that were exposed to 2.5 micromolars of cadmium. 6 that were exposed to 5 micromolars of cadmium. And finally, 6 that were exposed to 7.5 micromolars of cadmium. And then the sample numbers, they run from 1 till 30. And then what we need to put here for each of the reference genes are the RQ values that we just calculated. So I will go back to the tab with the reference genes and I will copy the RQ values. I will go to the input file for GreyNorm and then I will paste these values as numbers. Make sure to not just paste them using Ctrl-V, because in that case, Excel will not just copy the numbers, it will copy the formulas. And this is, of course, not correct, because then I will try to apply the same formula to this worksheet or this tab of the Excel worksheet, and that will obviously not work. So make sure to paste the numbers as numbers. So now we filled in the entire input file for GreyNorm. However, the GreyNorm algorithm will only function on a CSV file. So what we need to do next is we need to save the file as a CSV file. So I go to File, Save As. Then I look in my GreyNorm folder. I here click CSV, delimited, and I will give the CSV file a name, and I will call it input toxlearn. And it's very important that you save the CSV file in the same folder on your computer where the GreyNorm script is located. So I will save it. it will now tell me that for a workbook with several sheets, this will only save the active sheets, but that's what we want. We'll only save this sheet, which is our GreyNorm input file. So we just click OK. And then I will check in my GreyNorm folder, go to my documents, and then I here have my GreyNorm folder, and I can indeed see here the file named input toxlearn. Now we will need to run the GreyNorm algorithm. And if you want to use this, you can find all the required information on this website. So if you go to Google, you can fill in GreyNorm, and then it will lead you to this page. Here you can find all the files that you need to download in order to be able to use GreyNorm. And here you can also see that the software requires Python 3. So you need a platform to run this. So on this web page, you will find all the information on how to use the GreyNorm software. And then once you've downloaded all the required files, you can get started with it. So I will now go to my GreyNorm folder. And what I will do is I will click the GreyNorm file. This is a Python file. And normally when I double click it, it will immediately open it in the necessary software. Okay, there we go. And then the first thing I need to do before I can run the GreyNorm script, so at the top here, you can see the entire GreyNorm script. We'll drag this down a bit. You can scroll through it. But here in this entire script, you don't have to make any adjustments. So the script has been developed specifically to perform the analysis as we want. However, here at the top right corner, I need to change my working directory. So here I need to indicate to the software where I can find the GreyNorm script, but also where I can find the input file that I just generated. So I will click Change Working Directory, and I will go to my documents and then select the GreyNorm folder. I'll click Select Folder, and then here you can also see that now the working directory is correct, and it's indeed this GreyNorm folder. Then I will go back to the GreyNorm webpage, and then here under the title How to Use It, you find the line of script that you need to run in order to generate a GreyNorm output file. So I will just copy this line of text, paste it here, and then I still need to adjust two things. So what it will do is here, it will run the GreyNorm script. And then here in this piece, in this part here, you need to indicate the name of the input file. And in this case, the name of my input file was input toxlearn. And here in this last bit, you need to indicate the name of the output file. So I will name this output toxlearn. Then you just stand at the end of this row of text and you press enter. And as you see here in this folder, now the output file appeared. So what I will do is I will just copy all the data from this output file and put it in my original Excel file so that I have all the required information together in the same file. So now we successfully used the GreyNorm algorithm, and then you will see that we obtained quite an extensive output file. And what this algorithm, the GreyNorm algorithm, will tell us is which combination of reference genes is most suitable to use for the normalization of the data obtained from the gene of interest. And And the first column, you can see all possible combinations of reference genes. There are, of course, a lot of possible combinations because we measure the total of 10 reference genes, and it will test any possible combination of these 10. So it will test each of the 10 individually, all 10 reference genes together in one combination, all possible combinations of 2 genes, 3 genes, and so on. And it will rank them based on which genes are most appropriate to use for data normalization. And the ones at the top are the best to use. The ones at the bottom are the worst combination to use. However, remember from the lecture on data normalization that the golden standard is to always use a combination of 3 or more reference genes. And because of that, what we will do is we will scroll through the list, and the best combination to use is the first combination of 3 or more reference genes in the list. So according to GreyNorm, the best combination would be to use UBC and EF1A as a combination of reference genes. However, this is a combination of only 2 genes, so we will not use this one. The next one is to use EF1A as a single reference gene, but this is also something we will not do. But then in the next line, we find a combination of 3 reference genes. So this is the first combination of 3 or more in the list. So these are the 3 reference genes that we will select. Now I will give you a bit more information on what GreyNorm exactly calculates and what this ranking is based on. And the most important parameter that GreyNorm calculates, because this is also the parameter that the ranking is based on, is the so-called CV inter. CV stands for coefficient of variation, and this will tell you how much variation there is between the different experimental groups. So in the case of our experiment, how much difference there is in expression of these 3 reference genes between plants exposed to the different cadmium concentrations in the experiment. And here you can see that the CV inter is 0.13. But you will see as you scroll down the list that the values get higher and higher. And when we completely reach the bottom of the list, you will see here that the worst combination, or in this case it's a single gene, gives a CV inter of 0.57. So this means that it would not be a good reference gene because it shows a very large difference in expression between different experimental groups. And of course, the idea of a reference gene is that it shows very little differences in expression depending on experimental conditions. So then I will go back to the combination of 3 reference genes that we selected. And I will also explain to you what you can find in the subsequent columns. So in addition to the CV inter, so the variation between groups, it will also calculate the variation in expression, or in RQ values, within the different groups. So it will do this for, in this case, 5 groups, because we used 5 different cadmium concentrations. So you can see the variation within condition 1, which is the control, within condition 2, which were plants exposed to 1 micromolar of cadmium, within condition 3, condition 4, and condition 5. Then next, what it will also do is it will set the average in condition 1 at a value of 1. And then it will express the expression in all the other conditions relative to this first condition, which was the control condition. So you can see that if the average expression of these 3 genes in the first group, so the control group, equals 1, then the expression in condition 2 equals 0.77. So the expression is 77% of that in the first group. Then you can see in condition 2, it's 0.8. In condition 4, it's 0.71. And in condition 5, it's 0.76. And ideally, all these values should be close to 1, because if the values are close to 1, this means that there is little variation in expression between different experimental groups, which would make these reference genes good reference genes. And then you can also see that within each condition, the software also calculates standard deviation and the standard error. And then what it will also calculate is the average ratio of 1 over the normalization factor, which is the average value that you will multiply the RQ values of the gene of interest with in order to normalize them. So that's also something that ideally should be close to 1, because this would mean that the normalization would not have a strong impact on the obtained data. And then here it will also give the standard deviation for these values. So how much difference is there between the individual 1 over NF values. And it will also calculate a cumulative deviation from 1. So ideally, this is also as low as possible. But the most important conclusion from this GreyNorm output is that we will use a combination of SAND, UBC, and EF1A to normalize the data we obtained from the gene of interest, which we will calculate later. So then we will go back to the tab with the reference genes, and I will highlight the 3 genes that we selected. So SAND, UBC, and EF1A. It's these 2 and this one. So the only thing that we still need to do now for the reference genes is calculate this so-called normalization factor, which we abbreviate as NF. And the normalization factor is calculated by taking the geometric mean of the RQ values of the selected reference genes. The geometric mean in Excel, you can calculate it with the formula geomean. Then I open the brackets and I select the 3 values that I will use to calculate the geometric mean. So these are the RQ values of the 3 selected reference genes. And then I will close the bracket. And then I will drag this formula down, because we need to calculate the so-called normalization factor for each of the individual samples. And now this normalization factor is what we will then use to normalize the data of the gene of interest. So what we will do next is perform the calculations for the gene of interest. So I will generate a new tab, and I will call it SMR4, as this is the name of the gene of interest. Then I will again copy the first 3 columns of the first tab, which was the input data with the CQ values, and I will copy them here. So again, we have the 30 samples, the exposure group that they belong to, and then we have the CQ values of SMR4. Then what we will do now is exactly the same as we did for the reference genes, is that we will first calculate the minimum CQ. We again use the formula min, and then select all the data in the column, and then we obtain the CQ value for the minimum CQ value. So in this case, it's 26.68. So this is the lowest CQ value between all samples. Then next, we will calculate the so-called delta CQ. Oops. Voila. And the delta CQ is the difference between each CQ value and this minimum CQ value. And we then again place the $ sign in front of the row 34 to make sure that always this minimum CQ value is subtracted. And then we drag the formula down. And you can again see that the formula was used correctly because here we find a 0. And the 0 indicates that this is the sample with the lowest CQ value and hence the highest expression of the gene of interest. Next, we will calculate the RQ value, which is 2 to the power minus the delta CQ value. We press enter, and we again drag the formula down. So these are the so-called relative quantities, and they will show the relative expression of SMR4 in the different samples. So sample 21 is the sample with the highest expression of SMR4, and then the values in the other samples are expressed relative to the sample with the highest expression. For example, in sample 10, the RQ value is more or less 0.10, so it means that the expression in this sample is only 1/10 of the expression in sample 21. And these RQ values are the so-called non-normalized data. Now what we will do is we will normalize these data using the normalization factors that we just calculated based on the reference genes. So I'll add a column to include the normalization factor. I will go back to the tab with the reference genes, and I will copy these values that we just calculated, these normalization factors. And then I will paste them in this tab, but I will again paste them as values. So don't just copy paste with Ctrl+C and Ctrl+V, because in that case it will paste the formulas rather than the values. And here we need to have these values, because if you would paste the formulas, it would start using the same formulas on this tab of the Excel worksheet, and that would of course not be what we want. So we now have the non-normalized data, so the RQ values. Then we have the normalization factor. What we will calculate next are the so-called normalized relative quantities, or NRQ values. And we calculate these by dividing the RQ value by the normalization factor. Or you could also say that we multiply with 1 over the normalization factor. And then we again drag the formula down. So now we obtained the non-normalized data as well as the normalized data. What we will do next is we will calculate the averages of these. So I will here make a new title, non-normalized, for the non-normalized data. And what we will calculate for each of these are the average, the standard deviation, and the standard error. The average we will, of course, calculate from all samples corresponding to one experimental condition. So in this case, the average, just formula average, of, in this case, the RQ values, because we want to look at the non-normalized data. And we select the first 6 samples because all these samples belong to the same experimental condition, which is in this case the control. Then we also calculate the standard deviation from these 6 samples. So we type in the formula STDEV, open the bracket, select the first 6 RQ values, close the bracket and press enter. And then lastly, we calculate the standard error, which is the standard deviation divided by the square root, which is abbreviated in Excel as SQRT, of the number of replicates. And in this case, the number of replicates is 6, but this will be different for each experiment because maybe sometimes you will only have 3 or 4 or 5 replicates, or perhaps more than 6. So it's the standard deviation divided by the square root of the number of replicates, which in our case is 6. Then what we will do next is also calculate the average, standard deviation, and standard error for the other experimental conditions. You can do this by, again, filling in the formulas, but you could also just copy and paste the formulas to the correct lines. So always the first line of a new experimental condition. So here we now calculate the average, standard deviation, and standard error for in samples exposed to 1 micromolar cadmium, and the same for the 2.5 micromolars of cadmium, 5 micromolars of cadmium, and 7.5 micromolars of cadmium. Then in principle, it's perfectly fine to leave the data as they are, because what you can say now is that in the control condition, the expression is 0.17. Then after exposure to 1 micromolar of cadmium, the expression increases to 0.30, then stays around 0.30 for the 2.5 micromolars of cadmium, and then we see that it increases further for the 2 highest cadmium concentrations. However, by showing the data in this way, it's relatively difficult to say how strongly the expression increases, for example, between the control and the samples exposed to 7.5 micromolars of cadmium. So to make this easier to interpret, what I generally do is I calculate the non-normalized relative data. And for these, I again calculate an average, standard deviation, and standard error. So what I want to do is I want to convert this first value, so the average of the control condition, from 0.17 to 1, and then express all the other values relative to 1. Or instead of 1, you could, for example, also use 100, and then the values would be expressed as percentages. So what I will do to convert this value into 1 is divide the value by itself. And now, of course, we also need to divide all the other values that you can find here by this same value, because of course we don't want to change the proportions or the ratios between the different values. We just want to express them in a way that's more easy to interpret. So each of these values we will divide by 0.17. So what we can do is, because we always want to divide by the value in this cell, so cell I3, we will put a dollar sign in front of the I, so in front of the column, and also in front of the tree, in front of the row. And now we can drag this formula to the right, and we can also copy it to the different rows. And now it's much easier for you to interpret how strongly the expression of the SMR4 gene is increased by cadmium exposure. So we can see here that we have an increased expression of 1.80 times, 1.75 times, 4.6 times, and 3.9 times compared to the control and the different cadmium exposure conditions. And this is much easier to interpret than, for example, an increase from 0.17 to 0.66. Now we will do exactly the same thing, but we will do this for the normalized data instead of the non-normalized data. So we will perform exactly the same calculations, but this time using the NRQ values, so the normalized relative quantities, rather than the RQ values. So what we will do is we will again calculate the average, this time of the first 6 NRQ values. We will also calculate the standard deviation of these 6 values. And we will calculate the standard error by dividing the standard deviation by the square root of the number of replicates, which in our case is 6. And then we copy these formulas to the respective rows of the Excel sheet. And now we will also express all these values relative to the average of the control. And we do this by dividing each of the values by the average of the control, which is 0.32. And we here add the dollar signs in front of the row and the column to make sure that each value is divided by the value in cell Q3. And then we drag the formula to the right and paste it in the correct lines again. Then, to make things a little bit more visually appealing, you can also reduce the number of decimals in the Excel sheet. It always looks more neat and also easier to interpret. So now we calculated the non-normalized data as well as the normalized data. So what we will do next is we will generate a figure to visualize the data. And in this case, you can, for example, use a bar chart or a histogram. So I will insert a histogram. I will right-click and press Select Data. And then I will make two data sets, one with the non-normalized data and one with the normalized data. So I will fill in series name non-normalized data, and I will select the non-normalized relative values. So make sure you select the ones that are expressed relative to the control condition. And then I will add the normalized data. Again, the normalized relative data. Then I also need to change the axis labels because now my x-axis is just labeled 1, 2, 3, 4, 5. But instead, I want to replace this with the respective cadmium concentration. So I will select them here from my exposure column. And then click OK. So now you will see that the axes are correctly labeled. I will zoom in a little bit on the figure. So we can now click the figure, click the plus sign, and then add a legend. So now it will show that the non-normalized data are shown in blue. The normalized data, I will quickly change the name to make it look the same. So instead of normalized, I will write normalized data. Voila. So now we have in blue the non-normalized data and in orange the normalized data. Of course, you can change the colors as you wish. Here on the y-axis, you can see that there are too many decimal spaces because these zeros here at the end don't have much meaning. So I will remove them. So I will add here that I want to use only one decimal in this case. I personally prefer to also remove the grid lines from the graph, but these are, of course, things you can all change as you wish, according to your personal preferences. And then the last thing that I will do is I will also add error bars on the data. So what I will do is I will first select the non-normalized data. So I will select one of the bars and then automatically all of them get selected. I click the plus sign. I go to error bars, but I don't tick this box here. Instead, I click the arrow next to it and I use more options. Here you can then choose whether the error bars go both up and down or only one of them, whether you want a cap at the end or not. But this is, again, depends on your personal preference. I will then customize the values that I put in. So I click custom, I click specify value, and then I will select the standard errors, or if you prefer this, you could also use the standard deviations, as long as in a publication, in the legend of the figure, you indicate what the error bars represent, so either standard deviation or standard error. Then I will select the positive error values, so those are these. Make sure, again, you select those belonging to the relative non-normalized data. The negative error values are the same ones. And I click OK. Then next, I will also add the error bars in the same way on the normalized data. So again, make sure to use the relative ones. I select all of these. And now we also have the error bars on these data. Then here, I will still change some things about the axis. So now I see that the axis labels have changed a bit. So now the decimal numbers here at the end also don't really have any additional meaning. So I will remove the decimals so that we just have values ranging from 0 to 6. And then I will also give this axis a color. You see now it's white, but I will change it to black to make it clear that these are the axes. So I do this both for the x-axis and the y-axis. And now our figure is ready. So what you will see in blue are the non-normalized data. In orange, you see the normalized data. For the control, they have both been set at 1. And all the other values are expressed relative to these control values. And then you can see that with all the different cadmium concentrations, the expression of SMR4, the gene of interest, increases, but this effect is much stronger with the higher cadmium concentrations compared to the lower concentrations. And remember that what we will normally do is to perform the statistics both on the non-normalized and the normalized data, because the true expression lies somewhere in between, where we don't know. So this difference here between the blue and the orange bars is what we call the gray zone. And this is, this corresponds to the uncertainty in our qPCR data. However, even though here for 5 micromolars of cadmium, the gray zone is rather large, still we can say with certainty that after exposure to 5 micromolars of cadmium, the expression of SMR4 is increased, because both when comparing the blue non-normalized bars and the orange normalized bars, we see that there is an increase compared to the control. So generally, we will perform the statistics on both, so the non-normalized and the normalized data, and we will only indicate it as a significant difference in case it's significant for both non-normalized and normalized data. So this is what I wanted to show you in this demonstration. So you now know how to select reference genes using the GreyNorm algorithm, and also how to analyze data obtained through qPCR to calculate expression differences in certain gene of interest between different experimental conditions. And this is this method you can use for any gene of interest that you've measured.